There's a technology that is quietly, completely changing agriculture right under our noses. It's called KASP genotyping. And honestly, it's one of the most powerful tools plant breeders have for creating the tough, high yielding crops our world really, really depends on. So, how does it find that one perfect plant in a field of thousands? Let's get into it. All right, picture this. You're a plant breeder, and you're standing in a massive field. There are thousands, maybe tens of thousands of plants all around you. And somewhere in that sea of green is the one plant with the exact genetic trait you're looking for. Maybe it's resistance to a new disease or the ability to survive a drought. How on earth do you find that one special plant? That's the challenge. For years, the way we did this was, well, it was a total slog. The old methods were all about physically looking at plants, which was super slow, incredibly expensive, and you know what? A lot of the time, it just wasn't that accurate. But modern agriculture doesn't have time for that. What the world needs, is a system that's fast, cheap, dead on accurate, and can handle a massive number of samples all at once. So, here's what we're going to cover. First, we'll dive a little deeper into that needle in a haystack problem. Then, we'll introduce the solution, KASP technology. After that, I'll walk you through how its clever process actually works. We'll look at why it's so much faster, cheaper, and more accurate. And finally, we'll see the real-world impact it's having on the crops we all eat every day. Okay, so let's start with that core problem. What we're really looking for are these tiny, specific, valuable genes. We're talking about tiny variations in the DNA code called SNPs. That's for single nucleotide polymorphisms. The easiest way to think about them is like single-letter typos in the gigantic instruction manual of a plant's DNA. And just one of those typos can change absolutely everything. And this is where KASP technology comes in. It is just such an elegant and powerful solution to this genetic screening problem. Seriously, it takes what used to be a process that could take months and shrinks it down to just a matter of hours. So, what is this thing, really? Well, KSAPN stands for Competitive Allele Specific PCR, which, yeah, is a bit of a mouthful. But here's what it really boils down to. It's a super clever lab technique that makes two different versions of a gene compete against each other. And then it uses a fluorescent glow to tell you which one won. That glow tells you exactly which version of a gene a plant has. Simple as that. Now for the really cool part. How does it actually work? The true genius of KASP is just how simple and elegant it is at the molecular level. It gets you from a basic DNA sample to a crystal clear answer in just a handful of steps. So let's break that down. At a high level, it's kind of like a four-step recipe. First, scientists design these really unique DNA probes called primers. Second, they mix those primers in with the plant's DNA. Third, they stick it all in a standard lab machine that makes millions and millions of copies. And last, you just read the glow. You read the fluorescent signal, and you have your answer. Okay, here's where the cleverness really kicks in. The system uses two specific probes, almost like tiny genetic keys. One key is designed to fit only one version of the gene, let's say the version for drought tolerance. The other key is designed to fit only the other version. They literally compete to see which one can latch onto the plant's DNA. That competition, that's the K in KASP. Now, whatever key fits, the KCR machine starts making tons of copies of it. And here's the trick. Each of those keys has a little tail attached, and that tail is designed to grab onto a specific colored fluorescent dye. So if the drought-tolerant key fits, maybe the sample glows green. If the other key fits, it glows red. And that color tells us everything. Remember, plants get one copy of a gene from each parent. So if a plant got the same version from both parents, we call that homozygous, you'll only see one color light up, pure green or pure red. But if it got a different version from each parent, we call that heterozygous, you're going to get a mix of both colors. The color literally reveals the genetic code. The machine then reads these signals and plots every single sample on a graph that looks just like this. Each dot on here is one plant. And you can see it instantly, right? Three totally separate groups. You've got your homozygous plants for one version clustering together, your homozygous for the other version clustering down here, and all the mixed heterozygous plants sitting right in the middle. It's a beautiful, simple, visual answer sheet for some really complex genetic data. So we've seen how it works, which is cool. But why has this become the go-to technology for breeders all over the world? Well, the answer is a powerful mix of advantages that make it a massive leap forward from the old ways of doing things. The benefits are just so clear. KASP is ridiculously accurate. It's way faster and much, much cheaper than older tech. It lets you screen thousands of samples at once, which is huge. It's also flexible, so you can design a test for just one trait or a whole bunch. And, maybe best of all, it works on the standard lab equipment that most places already have sitting on the bench. And let's put a real number on that cheaper claim. Studies have shown that when you're using these genetic signposts to guide breeding, a process called marker-assisted selection, KASP can slash the costs by up to 44% compared to other big platforms. That is a massive, game-changing boost in efficiency. Okay, so this all sounds great in a lab, but what does it actually mean for us, for our food? Well, these advantages aren't just theory. KSP is out there right now being used to develop better versions of the staple crops that feed billions of people, making our food supplies safer and more secure. Take rice, for instance. KASPS is used to find a gene called SUB1. This amazing gene lets rice plants survive for weeks, even if they're completely underwater during a flood. I mean, that's a lifesaver in flood-prone parts of the world. It's also being used to find plants that can fight off rice blast, one of the most destructive diseases out there. And what about potatoes? KSP is a key weapon in the fight against some major threats. Breeders can now quickly find genes that give potatoes resistance to late blight. That's the same disease that caused the Irish potato famine. It also helps them find resistance to viruses and these tiny pests called cyst nematodes that can just destroy an entire harvest. For maize or corn, KASP is helping breeders get a step ahead of climate change. They're using it to find plants that have a natural genetic tolerance to extreme heat and drought. At the same time, it helps them find resistance to things like maize streak virus, which is a huge problem for farmers in sub-Saharan Africa. And finally, wheat. KSP helps on two fronts here. First, it can find genes that are linked to better quality for making bread, which is great for all of us. But it also helps find genes that stop the grain from sprouting on the plant if it gets too rainy during harvest, which can ruin a whole crop. It secures the food supply for the farmer. So KASP has totally changed the game for modern plant breeding. The speed, the cost, the sheer scale of it all. It lets scientists make smarter, faster decisions, and that means we get crops that are tougher, more nutritious, and higher yielding a whole lot faster. It really just leaves you with one exciting question. With a tool this powerful for reading the book of life, what incredible traits are we going to find next?